mentioned that last time, but it's important to mention that again. Just as the eversion system was the functional unit of compact bone, analogously, the functional unit of muscle contraction, what functions as one unit at a time, is the sarcomere. And to give you some idea of the minutia in terms of the scale of size here, what we're talking about, if we take the biceps and we bring it down to the cellular level, the fiber level, and look at the individual muscle cells, which there are millions in the biceps, and we take one of those muscle cells, one fiber, that contains over 100,000 sarcomeres in one muscle cell. Okay, so you can see the magnitude of the contraction mechanism that occurs in terms of the multiplicity of sarcomeres that are found in the body. Now we're taking the biceps, which is a particularly very heavily contractile muscle in terms of arm movement, which we're doing a lot of. So you'll see probably more sarcomeres in a biceps muscle fiber than you would in some of the less active muscles in the body. But that gives you some idea of the scope of how many sarcomeres are involved in getting muscles to contract. So let's now go down and look more in depth at that fifth level of organization and see how an individual sarcomere is organized. We talked a little bit about this last time because we did talk about the mechanisms. But here's the layout. Here's what you see under the microscope when we looked at the striations and the banding patterns of the skeletal muscle. And we have some definitions here in terms of some of the terms that we'll be using as we describe muscles that you need to familiarize yourself with and where they are in the sarcomere and how the sarcomeres line up. Basically, sarcomeres line up in repeating patterns. So we have one after another. They are kind of like building blocks of proteins, like the amino acids that lined up in proteins in sequence. Well, sarcomeres are all the same, but they line up together so that as they shorten, they contract, and as they lengthen, the muscle relaxes. So when we look at the individual sarcomere, it's structured by what we call a Z-disc. And this is a three-dimensional kind of a protein that is the border of each and every sarcomere. So the sarcomere is defined, the functional unit is defined as the length of one Z-disc to the other. And these Z-discs will line up ad infinitum in terms of the cascade needed to fulfill one muscle fiber's cell of 100,000 sarcomeres. So they line up together in repeating patterns. And the sarcomeres are made up of the thick filaments and the thin filaments. The thick filaments are the myosin molecules, contractile proteins, and the thin filaments are the actin contractile proteins. And it is their interaction that causes the muscles to contract. So I've scaled it out for you on the board here by these very thick blue lines being the myosin filaments and the very thin green lines being the actin filaments. And we know from our discussion on Monday that it's the interaction areas where the myosin and the actin overlap, where we have the actual formation of the cross bridges and then ultimately the power stroke, which will tweak the sarcomere and shorten the sarcomere, and contraction will occur. So in this particular instance, we have the thick filaments that line up along the sarcomere, and we have individual thin filaments. And the thin filaments, as you can see, extend all the way from one sarcomere to the other, and there's a small period of time when there are no thin filaments. There's a larger period of time, or a larger length, between sarcomeres from one to another, where there is actin but there is no myosin. Okay? So what we have is the borders of these proteins, the Z-discs, that border the sarcomere, and we have the myosin and the actin, and points of in between. Now there's a point in the middle of the sarcomere which we call the M line, and that's basically the middle line. So that's 50% on the left and 50% on the right of the sarcomere. And bordering in that midline is an area that we call the H zone, or sometimes referred to as the H band. And the H band is simply defined as the middle region of the sarcomere, and in the middle of the H zone is the M line. So the M line is midpoint of the sarcomere and also midpoint of the H zone. In the H zone, you'll notice that the thin filaments are missing. So it's just the myosin, the heavier filaments. And when you see the banding patterns under the microscope, what you look for is the dark bands and the light bands. And where you see the dark bands is where we have the overlapping of the actin and the myosin, because there's more concentrated material there. So you'll see a dark pattern, then you'll see a light pattern in the middle, then you'll see another dark pattern, and then a fairly longer light period, light pattern between the Z, from one end of the Z disc to the beginning of the other Z disc. So the H zone is simply defined as the middle of the sarcomere, and it's also the area where the thin filaments are absent, so all we have is myosin. The A band is the band that runs the entire length of the thick filament. So the myosin from one end to the other is all concentrated in what we call the A band, the entire length of the thick filament. The I band is a little bit more difficult in concept because it runs between sarcomeres. So the I band is between the Z disc and the ends of the thick filament. And the ends of the there are no thick filaments in the I band. So just as the H zone had no thin filaments, we see the I band has no thick filaments. Okay, so the actin runs all the way through the Z disc and into the next sarcomere. Right?
Thank you.
So what we want to do is we want to define these areas, and we want to watch and see how they change during contraction and relaxation. Do they shorten? Do they lengthen? Do they stay the same? What happens when the muscle contracts to the I band, to the A band, to the H zone? What happens then when relaxation occurs? Okay. So this is our layout in terms of our definition. Now let's pick up on the ability of the muscle to contract. So if we take what we learned on Monday in terms of our mechanism of action, molecular mechanism of action of the contraction mechanism, what would you say about this particular muscle?
Relaxed.
It's relaxed. Very good. Why would it be relaxed?
Because
They're not attached.
the heads are not attached to the actin.
The heads are not attached. Okay.
Actin filament.
In normal resting pattern, normal relaxation pattern, what do we see? We see the myosin heads pretty much lying at a
45.
very slight angle, 45-degree angle, up on the myosin, but not touching the actin heads, right? And we know that the actin has binding sites, right? And these would be very close to the myosin heads. And in relaxation, the myosin heads are not bound to these actin binding sites. So very good, you did determine that this is a muscle in relaxation. Now, why are these myosin heads not bound to the actin binding sites? So the blocking protein, right, which was what, the troponin? And the tropomyosin complex, which actually is two molecules. And that blocking protein, troponin and the tropomyosin, is sitting on the actin binding sites, right? So this complex in relaxation sits on the actin binding sites and blocks them. So contraction cannot occur. Okay? So in order for the contraction to occur, what has to happen? Where do we have to go to find the answer? How are we going to get this
neurons.
blocking protein off of the actin binding sites? What's the chemical signal? What's the molecular mechanism? Where are we going to go look for that?
From the neurons.
At the neuromuscular junction, right? And what is the neuromuscular junction? Anybody? Catherine? Catherine, you didn't raise your hand. So the axon terminal
Right.
is one part of it, right? That's the neuro part.
Then you have the end wall, so it's that piece